Alright guys, so we're going to get started here, and what I'm going to do is basically present to you what I've been doing at the university for my research. So a lot of you guys are curious about what I do and where I work, and I'm going to take this opportunity to share that with you. And I'm going to start off by presenting a PowerPoint quickly. So what we're interested in finding out is when people take traditional medicines, they often think about traditional medicines as being this foreign thing with a lot of weird plant products, or maybe they're thinking about things in glass jars, but the reality is we all take traditional medicine sometimes. Things like grapefruit, garlic, ginger, ginkgo, or St. John's wort are all traditional medicines that we often use if we're not feeling so well. So there might be a time where yourself or maybe one of your parents is feeling really sick and they're having some sort of herbal tea that has like lemon or something else, and they also take just regular medicines. And sometimes when you take both of those things, traditional medicines and regular medicines, they don't work. And you have a really bad time and you're frustrated and you're still sick, and it's because you're taking both of those medicines. So, not always, but sometimes, if you take traditional medicines and regular medicines together, you're going to have a bad time. So, what we're interested in is we want to find out when medicines are not working properly. And in order to do that, we are studying a molecule called cortisol. And this is just a really simple kind of cartoon animation of what that molecule looks like. Now, that molecule, cortisol, floats around in our bloodstream, and it's bound up by a protein called corticosteroid binding globulin, or what I call CBG. So CBG is the protein that carries cortisol around the body. And remember, we've learned about proteins, and we know that proteins are the molecules in the body that do all of the work. So here's an animation, or just a quick picture of what the protein looks like. And you can see the arrow is pointing to the molecule of cortisol. So, we want to know when medicines are not working properly, like when you're taking traditional medicines and regular medicines together. We want to measure the molecule cortisol, and we want to do that by using the protein corticosteroid binding globulin. Now, you're probably wondering, how on earth do you make a protein to study or use in an experiment? And it's actually pretty simple, especially considering what you guys have learned so far this year. So if you remember the bacterial cell, inside of the cell we've got all this DNA that's packaged inside of the chromosome, and that's all the information. And then if you look, some of you guys notice this, there's this thing called the plasmid, and it's also DNA. So what we can do is we can take DNA that we want to study, and we can put that DNA, the information molecule, inside of the bacterial cell, and that bacterial cell can then start making our protein. So I know it's a lot to digest, but we're going to watch a couple of videos here, and you guys are going to have the opportunity to ask me questions if you don't understand. So I hope you're ready to start watching these videos.
Get ready for a lot of fun and excitement. Now, let's start.
So here we are in the lab of Dr. William Folk, where for the past 3 years I've been working on this protein called CBG, corticosteroid binding globulin. This is the bench where I do most of my experiments, but not all of them. And if you take a look around, you can see that there's a lot of things. And all year long I've been harping on you guys to make sure that you're extremely labeled and extremely well organized. And this is why. As you can see, there's a lot of things, and experiments in science can get kind of confusing. So it's really important that you label clearly and stay organized. So let's get started with the first part of the experiment, the heat shock. So now that we've got our ice and our bacterial competent cells, BL21, we're ready to start our heat shock transformation. Now, if you guys remember from your mini grant research, bacteria really don't like to grow or do really anything when it's really cold. That's why we're keeping everything on the ice for right now. Inside of this tube, I have the CBG, the DNA from a rat CBG, and we're going to try to put this inside of these bacterial cells, BL21, using heat shock transformation. Now what that means with competent cells, when you put them in a hot water bath where it's really hot, the cells start to swell up, leaving holes inside of the cell. Once that happens, the DNA can enter the cell, and then when we put it back on ice, the cells shrink up again. And now our DNA is hopefully trapped inside and the bacteria can start making our protein. So in this step, we're going to add our DNA to the competent cells. Now, we also have a control here. Instead of adding DNA to one of the tubes of competent cells, we're going to add water, and water only. Now, this is a negative control. If we only add water, we shouldn't expect to see any bacteria grow up. So here I'm going to take the CBG DNA from rat, and I'm going to take 20 microliters. It's a really small amount. And I'm going to quickly add it to these competent cells. I'm also going to do the same thing to the sample of water, the negative control. After I've added these to the tubes of competent cells, the next step is to let them rest on ice for a few minutes before we do the heat shock transformation in the hot water bath. So here we are by the water bath. There's a lot of different water baths in this lab. This water bath is set at 37 degrees Celsius. This water bath here is set at 90 degrees Celsius, but the one that we're going to be using is this water bath here, and it's set at 42 degrees Celsius. Now, what we're going to do is we're going to take our competent cells and we are going to stick them in the water bath for exactly a minute. And because the water is really warm, those cells are going to expand and holes are going to open up in the bacterial cells, and our DNA will be able to get into the bacterial cells. After a minute has passed, we are going to take the cells out of the water bath and stick them back on the ice for 5 minutes. When they're on the ice, the cells are going to shrink up again, and hopefully our DNA will be trapped inside. The next step after we do that, after they've been resting on the ice, is we're going to add those competent cells to tubes of broth, bacterial broth. You guys remember this. We've been working with bacterial broth a lot. So this is just the liquid that has all the food and all the nutrients that the bacteria need to grow. When I add the bacteria to the broth, they're going to go into the 37-degree water bath for about an hour or two, and then we're going to put them on a plate overnight and see what grows the next morning. So after the heat shock transformation and the cells having been incubated for over an hour at 37 degrees, the next thing for us to do is to plate these onto LB agar plates, just like the ones that we've used in class before, and grow them up overnight. Now again, I have 2 plates. I have one for the stuff that I want, the CBG inside of the bacterial competent cells, and then I have another one for the negative control. Now you should remember from experimental design that on the negative control, we're not expecting to see anything, and that tells us if our experiment is working or not. So since I'm not working in a biological safety cabinet and I'm working at the bench, it's really important that I keep everything sterile. And so for that, that's why I have this burner. Most lab benches have these gas valves. So I can turn on the gas. I can use this to make a spark. And there we have our flame. The next thing we need to do is to take this glass spreader. We're going to dip it in some ethanol. And then we're just going to heat it up to kill any bacteria that might be on the spreader. I'm going to take a sample of my cells. put it on the plate, spread the plate, and then put it in the incubator overnight. And the reason that I'm working by the flame is because the air around the flame might contain a lot of bacteria, and the flame should be able to kill off any bacteria that's floating around the plates. So there are a lot of incubators in this lab, but for growing up these plates overnight, we're going to use the 37-degree incubator, and it's right here. We've got our 2 plates, again, CBG and our negative control. Now remember, whenever we store these, we store them upside down so that we don't form moisture on the top of the plate. We're going to stick these in here overnight and take a look at them tomorrow morning to see if hopefully nothing grows up on the negative control plate and we see a lot of little colonies on the CBG DNA heat shock transformation plate. Alright, so if you guys have any questions, we can probably take a few minutes of questions.
Okay, can you see us yet?
Yes.
You can see me?
Or I can't see you guys, but I can hear you.
Okay, well, I'm going to let the kids come up one at a time to the microphone and ask a question, okay? Mm-hmm. Okay, EK? You don't do anything but talk.
Where's the microphone?
You don't have to do anything but talk to him. Say, hello, Mr. Alam.
Hello, Mr. Alam.
It's EK.
Hello, EK.
It's EK. When you burn that stick, what are some bacteria that do very well with fire?
So 99.999% of bacteria are not going to be able to survive getting flamed through the fire. So that usually kills off any bacteria.
What about the other percent?
Usually they just don't grow up. So it should kill everything. I can take another question.
This is Sydney. Did you do this video before you came to us? Or we got the mini grant, right? And then you did the video and then we started our experiments? Is that what happened?
So the question is about when I filmed the video, whether it was before the mini grant or after the mini grant. And the video portions were actually filmed in March, so after we had started our mini grants experiments. I'll take one last question and then we'll go on to the next video.
Will they get a chance to ask questions some more?
We'll have a little bit of time after this next video and after the last video to answer some more questions.
Hi, Mr. Alam. You know
Hi,
who I
do you
am?
have a question?
Yeah. What was that thing
Go
that
ahead.
you were scraping with the agar?
So that thing is called a plate spreader, and the question was, what was that thing that I was scraping on the plate? And it's basically just a plate spreader. I can put bacteria on the plate and spread it all the way around the plate so that it grows up evenly and not just in one spot.
Why didn't we use that?
We didn't use a plate spreader because there was no reason for us to use a plate spreader. We didn't have a tube full of bacterial cells anyways. So, with that said, we're going to go to this next video. Alright, now that our plates have incubated overnight, it's the fun part. It's time for us to see if we've actually gotten any results. And this is the most exciting part for any scientist, to see if things actually work. Sometimes they don't work, and it's pretty depressing, but that's science. You just kind of have to move on. Now, if you remember, we've got 2 plates. We've got a negative control, which we transformed it with just water, so there shouldn't be anything on that plate. And then we also have our plate with our CBG-containing bacteria, hopefully. So let's take these plates out, and then we'll check them out on the light box to get a closer look. So let's take a look at our plates. So here's our negative control, and thankfully, absolutely nothing has grown on this. And what that means for us is that our experiment is working as it should. Now, moment of truth. This is the plate with the CBG transformed into the competent bacterial cells. As you can see, there are a lot of colonies on this plate. So this experiment has worked out really well for us so far, and now it's on to the next step. So the next step in the process is we are going to take a colony from this plate and we are going to grow it up overnight in a tube of broth. That way, tomorrow morning we'll have a lot more bacteria, and then we'll continue to keep growing up that bacteria until we have a lot of bacteria. That way, since our bacteria is going to be making our protein, we'll have a lot of protein so that we can study it even more. So the first thing that we have to do, again, is I've got to turn on this gas valve. I'm going to use the fire starter to give us a nice flame such that we can have a sterile environment. And what I'm going to do is I'm going to take a colony from this plate and I'm going to mark it on the plate by drawing a circle around the colony so that I know, for future reference, which colony I have actually taken from the plate. I'm going to make sure to work by the fire. Again, the fire, we're hoping, is killing all the bacteria that's in the air so that bacteria we don't want doesn't grow up. I'm going to take one of these tips. I'm going to lightly just scratch the surface of the agar and try to get some of that bacteria onto it. And then I'm going to take the bacteria that's on this little tip and I'm going to dump it into this tube of broth. Now we've got bacteria to grow up overnight. So this is another style of incubator that we have here at the lab. There's 3 of them here, and we're going to use the one at the middle, because the one in the middle is set at 37 degrees Celsius, which is the optimal temperature for this bacteria to grow up. Now, this incubator is a little bit different from all the other incubators, because this thing rotates, so it actually shakes the sample up. Alright, do you guys have any quick questions before we get to the last video?
Mr. Alam.
Say, this is Zaccharia.
Hi, Mr. Alam. I'm Zaccharia. Okay.
And your question is?
But how did you know? Wait a second. He made this video in March. Okay, so.
Quick.
I don't got no
Okay,
question.
go ahead, AJ. Sorry, Zaccharia forgot.
Mr. Alam, this is AJ. I have.
Hello, what's your question, AJ?
Did you really use rat DNA?
Yes, so the question is whether or not we actually use rat DNA, and we actually do use the rat DNA and put it inside of a bacterial cell, and that bacterial cell starts making a rat protein inside of it. Okay, let's do one last question and then we'll get to the third video.
Hey, Garrett, move it quick.
Hi, Mr. Alam. I'm Garrett. How hot is 30 degrees Celsius? 37 degrees Celsius?
So the question is, how hot is 37 degrees Celsius? And it's in Fahrenheit, it's body temperature, so it's 98 degrees Fahrenheit. So when we've grown up bacteria in the classroom, we've used that exact same temperature, 37 degrees Celsius or 98.6 degrees Fahrenheit. So I know you guys have a lot more questions, but we're going to watch one last video, and then we'll take as many questions as we can, and anything that we can't address today, we can address in class the next time I'm there. So here we are on day 3 of our experiment. We've got a lot to do today. We've got to express and purify our protein from our bacterial cells. But before we do that, I thought it was probably a good idea to review what we've done so far. So remember that our goal is to produce this protein, CBG, in bacterial cells. Now we use bacteria because, as you guys know, bacteria grow up really fast. They're also really easy to work with and can make a lot of protein for us. So, on day 1 of our experiment, we did a heat shock transformation. That is, we took the DNA, or the information for CBG, and put it inside of bacterial cells. We did that by placing the competent bacterial cells into a hot water bath, and that caused those cells to expand and form holes on the outside of the cells. And that allowed for our DNA to get trapped inside of the cell. When we put those cells back on ice, they shrink up and now our DNA is stuck inside of the bacteria, and the bacteria will start making that protein. We plated those cells onto LB agar plates overnight, and the next day we had a plate full of colonies. We took one of those colonies, added it to 10 milliliters of bacterial broth, and let them grow up overnight. So today we're going to start with our 10 mils of overnight culture. And although it's a lot of bacteria, we need a lot more bacteria if we want a lot of protein to work with. So we're going to take that 10 milliliters of bacteria and grow it up into a liter. Then we're going to collect all of those bacterial cells and we're going to burst them open because our protein is trapped inside the cells. So the only way we can get our protein out of those cells is if we burst them open. So we're going to be using a sonicator to do that. The next thing that we're going to do is, when we burst open those cells of bacteria, we can get all the protein out of them. And CBG, the protein that we want to study, isn't the only protein the bacteria make. The bacteria make hundreds, if not thousands, of other proteins. And so in order for us to get that CBG protein, we're going to have to purify it out of the sample. And once we have our purified sample, we're going to check to make sure that it's pure, that we actually have the sample that we're after. So, it's going to be a long day, there's going to be a lot of experiments, and I'm hoping you guys can help me out. So here's our tube of overnight culture, and I'm hoping that you guys can notice a huge difference from the before to now. You can probably tell that this overnight culture, after it's been incubating overnight at 37 degrees Celsius, is a lot more cloudy. And that's because we went from just one colony inside of the tube, and it grew up overnight. So we went from one, and we probably now have millions, if not billions, of bacteria inside of this tube. Now that might seem like a lot, but if we want a lot of protein, we actually need to grow up a lot more than just 10 milliliters. Here we have our 10 milliliters of overnight culture. We've got a couple other things here as well. I have this giant 4-liter flask that we're going to use to grow up a liter of bacteria. And here's a liter of the LB solution. So this part's pretty easy. Let me get some gloves on and we'll get started with it. So what we're going to do is pretty simple. Inside of this flask, we're going to add our 10 milliliters of bacterial broth. We're also going to add 1 liter of fresh, sterile, autoclaved LB broth. As you can see, we're not going to be able to fill up the whole flask, but that's good because we want to make sure that the bacteria have enough oxygen, which is why we use such a large flask. So that's it, pretty simple. We're going to put this outside back into that same 37-degree incubator that rotates these 250 times a minute, and we're going to let them grow for about 6 hours. After about 6 hours, we should go from this relatively clear solution to a really cloudy solution of bacteria like we just saw. We're going to have probably trillions of bacterial cells, and we're going to need to collect them and burst them open for the next part of the experiment. So 6 hours have passed since we put our overnight culture into a larger 1 liter of broth. Notice anything different? If you look at the before picture, you can see that when we started, the broth was pretty clear because it was fresh, it had just been autoclaved, and there was absolutely no bacteria growing in it. We took our 10 mils of broth, added it to this 1 liter, and 6 hours later, we've got billions, if not trillions of bacteria inside of this flask. So the next thing that we have to do is we need to get rid of all this broth, and we want just the bacterial cells. So we're going to use the centrifuge, and what that's going to do is it's going to separate everything that's heavy from all the things that's light. And what that means is that all those bacterial cells that are just kind of floating around in this broth are going to get pulled to the bottom of a tube, and all the broth is just going to stay on top. That way we can pour off all the broth and we'll just be left with our bacterial cells, and then we'll be able to sonicate them. So this is the centrifuge that we're going to be using to spin our cells and to collect our cells. Now, I've already transferred our bacterial broth into these containers that are meant for the centrifuge. Now, when we put them inside the centrifuge, they're going to spin around in a circle at about 10,000 times the force of gravity. So if you weigh 75 pounds and you were spinning around this fast, you would actually weigh about 750,000 pounds. So it's a lot of force, and that force is basically going to pull all the cells that are floating around down into the bottom of the tube. Then we can pour off all the used broth, and we'll have just our cells in a nice little pellet at the bottom of the tube. So this is going to run for about 10 minutes at 10,000 times the force of gravity. After our cells have been spinning for 10 minutes, we can take them out and take a look at them. We'll see that we have now a nice pellet of cells at the bottom of the tubes. Perfect. Now we can pour off all that extra broth from the top of the sample. And all we're left with is that pellet of cells. So here we are by the sonicator, and the sonicator is actually this tall thing here, and the unit that controls it is right here. And the way this works is a sonicator is called a sonicator because it uses ultrasonic frequencies. So basically the tip of the sonicator is at a frequency so fast that it just kind of causes all the bacterial cells to just explode, to burst open and to spill all their guts, essentially. And so we're going to be using the sonicator to open up our bacterial cells so we can take CBG out. Now, I've collected all my cells and I transferred it to a different tube so that it's easier. I've got my bucket of ice once again, and we're going to be working on ice while we sonicate because we want to make sure that everything stays nice and cold and that our protein doesn't degrade because of the temperature. So we're going to go ahead and use this, and you're probably going to see me putting these things on when I'm sonicating because these ultrasonic frequencies can do some damage to our ears as well. So it's important that I protect myself from the sonicator. The bacteria, not going to be so lucky though. So I've got my sample. It's in ice, and I'm going to turn on the control unit for the sonicator, and we're going to be sonicating it for 30 seconds at a time. We're going to do it 5 times, and we're going to let the cells rest on ice for 2 minutes in between each sonication step. So we're almost ready to start purifying our protein. But before we do, we've got to do another centrifugation step. Now, instead of spinning the cells to collect the cells this time, what we're going to do, since we've already sonicated the cells have exploded, and our CBG protein is just kind of floating around. What we're going to do now is we're going to centrifuge it so that we can collect all the things that we don't want. Things like the cell wall and the membranes and all the other things that weigh a lot. So we're going to separate all that heavy junk from the cell, the cellular debris, and hopefully at the top of the sample is just going to be what we call a supernatant, or the soup. And that soup is going to contain our protein, and that's what we're going to be purifying. So about 10 minutes have passed now. Let's check out our sample. Now, if you remember the way it looked before, it's probably going to look a little bit different now. So you can actually see, if you look up close to it, all the cellular debris, the cell membrane, the stuff that makes up the cell wall, and all that other gunk is now at the bottom of the tube. And at the top of the tube, sort of at an angle, we have our supernatant, our soup, or the stuff that contains all the proteins inside of that cell. Since the cellular supernatant, or the stuff that's floating on top of the rest of that cellular debris, is the stuff that we want, I'm going to transfer it to a new tube, and we're going to be ready to start our purification process, where we separate the CBG protein from all the other proteins that the bacterial cell makes. If you remember, we were started with a liter of cells, and this is all that we have left, is this little tiny amount inside of this tube. And inside of this tube, there's a lot of different proteins. CBG is going to be one of them. So you can see why we needed to grow up a larger sample just to get this small amount. So this is how we're going to be purifying our protein. This thing is called an affinity chromatography column. And although it's a big word, it's pretty simple. The gel that's in here at the bottom of this column is going to bind and grab onto our protein inside of this tube. And only the protein that we want, our CBG. Everything else, when we pour it in, is going to flow right through the bottom. And I've already done a lot of the prep work, but if I were to just add a little bit of some solution to there, you can see that the solution starts to drip out from the bottom of the tube, which is perfect for us. Everything except for our protein is going to drip out of the bottom of our tube. So let's load in our protein, and we're going to let this column incubate with the protein for a little while. And after probably about half an hour, we're going to add a chemical to the top of the column that's going to kick off our protein. So our column has been incubating with all the proteins, including CBG from the bacterial cell. It's been about 30 minutes, so now we can uncap this and let all the proteins flow through. You can see at the bottom of the column, all that supernatant is going right through the column. We're going to wait for a little bit and make sure this all goes through, and then we're going to wash the column with some solution just to make sure that we get everything off of it except for our protein, and then we're going to be on the last step where we add our chemical to the column and it kicks off our protein, and we'll collect that protein inside of a tube, and we'll be ready to study our protein, to do whatever we want with our protein. And And that's what I'm going to need your guys' help to help me figure out how much protein I actually have. So after we've washed our column a few times, we can move on to the next step. And in this step, we're going to add the chemical called glutathione to this column. And that chemical is going to kick off all the CBG protein that should be stuck in right here. And you can see underneath I've set up a tube that's going to collect all of my sample. Probably a good idea that I actually lower it to make sure I don't miss any of my sample. So I'm going to add my glutathione to the top of the column. And everything that comes out of the bottom should be my protein, my purified protein. And again, when I say purified, that means that there are no other proteins in that sample. There's a lot of proteins that bacteria make, so when we busted open that cell, there's probably thousands of different proteins. And we only want what's stuck on the column here, which is our CBG. Alright, so I know that was a lot for you guys to follow, but if you guys have some questions, we probably have about 5 minutes to take those.
So you started off with that little tiny bacteria cell. Is that what it was? What was it?
So the question is that, did I actually start off with just that little tiny bacterial cell? And that's right, we start with one cell and we grow it up to billions and trillions of cells, and we're able to get the protein that we're making out of those cells.
Okay, so that's pretty amazing to go from one cell to trillions of cells. Okay, Daniela?
This is Daniela, and I was asking, why did you use the giant triangle tube instead of a small one?
So the question is, why did I use the giant triangle tube instead of a small one? Is that right?
Yeah.
Okay, so that giant triangle tube is a thing called a flask. And we used such a large flask because we wanted to grow up so much bacteria, and we needed a lot of broth to grow up all that bacteria. I hope that answers your question.
Okay, here comes Will Do Lady.
Hi. Alright, so how come you had so much of that, I think it was the broth stuff, and then you had like a teeny tiny little bit of it left, I think.
So the question is, how come I started off with so much broth and just ended up with such a small amount of stuff? And the reason is because we grow up all that bacteria, and we need a lot of the broth to do so, to grow up all that bacteria. But each one of those bacterial cells is really, really, really small. So when I used that centrifuge and spun down the sample, all those little cells went to the bottom of that tube and just formed that little clump of bacterial cells. There's probably trillions of them there.
Okay.
Okay, anybody else have a question?
Oh, I have another
Somebody
one.
has not been up there.
I have
Okay,
another
Zakira,
quick
you have
one.
one?
I have
She didn't ask a question. Go, Zakira.
Hello, Mr. Alam again. I got a question. How come when you put that gel in that thing, how come it didn't come out?
So I'm not sure what the question is, so can you repeat that?
I said, how come when you put that gel in there with the solution, how come the gel didn't come out?
Okay, so the question is, how come when I put the gel probably inside of that column in that solution, how come it doesn't come out? Because the bottom of that column has a little filter it's kind of like a coffee filter, and it makes sure that the gel isn't able to pass through. It's kind of like a strainer for spaghetti in a sense. So we'll take one last question.
No, Will Campbell now.
Hello. Can I ask 2 questions?
Go quickly, Will.
Just ask one question. If we have time, we'll do the next one.
How come you use rat DNA instead of human DNA or something?
So the question is why we use rat DNA instead of human DNA. And the reason's a pretty simple one, and it's because we had the rat DNA available to us. And it's as simple as that. So what's your next question?
Do you have another question, Will?
I don't remember the last one.
Alright guys, well I hope you enjoyed the research broadcast, and if you guys have any questions, write them down in your science notebook, and the next time in class you guys can ask me whatever you'd like to.
Okay, thank you, Mr. Alam.
Bye, Mr.
Bye.
Bye!